dear colleagues, let us come to our final talk today, and for final talk for the whole conference. But the point is, the next speaker was invited the first. The first person I invited for this conference was our next speaker. And it's going to be Michalis Averof. And we have a long history with Michael, because when I was finishing my PhD in Japan, I thought, oh my God, I want to work in something exciting. And I found his studies on evo-devo, on evolutionary genomics. And I wrote to him like asking, let's think about doing something together. And he said, maybe not easy to find here in Japan and so on. So it didn't work at all. But it stimulated me when I found the topic I worked for the last 10 or 12 years, because I thought, it's so exciting. Not all model organisms are cool, so something really exotic and new models are very important. And today it's my great pleasure to introduce Michalis Averof, the member of Institute of Functional Genomics in Lyon, with a talk Beyond Model Organism, from evo-devo to regeneration. We have very nice regeneration line of talks today. Michalis, are you with us?
Yes, I'm with you.
Thank you.
Thank you very much for the invitation and the introduction. Can you see my slides?
Now you will start to share and probably will be okay. So please start to share it. Yes, very nice.
Yes? Okay. Okay, so thank you very much. Oleg and Igor, when they invited me, they asked me if I could give a general perspective on the questions and topics that have interested our team over the years. And for this, I would like to start with this picture, which highlights the contrast between the diversity of animals that we find in nature and the a handful of model organisms from which research has given us information on this diversity. So the contrast here is that in the animal kingdom we have a huge diversity in morphology, physiology, and behavior. And almost everything that we know about the genetic underpinnings of that biology comes from a very small number of model organisms. The justification for this is not that these organisms are particularly representative of this diversity, but there are two reasons why we do this, right? The first one is that in these animals, research communities have developed very extensive genetic tools and technologies and approaches through which we can study the genetic underpinnings of development or physiology or behavior. And the second reason is that is what Monod used to say, that what we learn from E. coli will apply to the elephants. Is that much of what we learn through these model organisms has actually a much broader applicability to the whole animal kingdom. Our team has been interested in this contrast and trying to find ways to escape the kind of very strong focus on model organisms and try to study some of that broader diversity. And I think there are two major justifications for wanting to do that. At least there have been two major justifications for this in my mind. The first one is in order to understand evolution, in order to understand the historical events and the mechanisms of animal evolution. It is necessary to go into the past. There are two ways really we can do that. One way is to look at fossils, look at what remains from the past, and the other way is to do comparative studies, to take the diversity of animals that we see today, compare them, and through these comparisons to try to infer what was happening in the ancestral states. And my own initial entry into biology and into research had been exactly in this domain, what at the time was the beginnings of the discipline of evo-devo, studying and comparing the role of Hox genes in different parts of the body, in different arthropods, in order to try to understand the evolutionary changes that have taken place in different groups of animals and the possible mechanisms and genetic controls of these changes. Since then, of course, evo-devo has established itself as an entire discipline, and there is a very interesting component of evo-devo that looks, rather than looking at the macroevolutionary changes that I was pointing to earlier, is looking at the micro scale, making comparisons between either very closely related species or races, or even domesticated varieties of plants and animals, in order to understand really the details of the genetic architecture that underpin morphological change during evolution. So the first justification is really to understand evolution. We need to look broader than model organisms and have a comparative approach, whereby we have to get into the study of the genetic underpinnings of diversity through different species. The second one, which has not been so explicit, I think, is that model organisms tell us a lot about what is going on in most animals, but they do not represent every aspect of biology that we have in the animal kingdom. There are very exciting biological processes which are simply not represented in this handful of organisms that we use as models. And I want to give just a very quick example here to indicate that this new biology that I think we can discover in new species is not merely minor variations in the themes that we already know, but they can lead to really very different ways of understanding the biology of some animals. So the example I want to give is this process which used to be called chromatin diminution when it was first discovered. Now it is more commonly called programmed DNA elimination. During development, as cells divide, we are used to the idea that chromosomes are properly segregated in the progeny, and that every cell that is generated from the fertilized egg will carry the same genome, the same genetic instructions during development. And yet in some species, there are particular cells which do something very striking. They break their chromosomes into hundreds of bits. So this is what you see here. This is a nuclear stain. This is a DNA stain in a single cell of ascarid nematode. And what you see is that the DNA has been broken into hundreds of pieces, and then during the segregation of the genetic material during the subsequent division, what actually happens is that a small part of the genome is segregated to the daughter nuclei, but another very substantial part of the genome stays at the center and is eventually lost. It's eliminated from these cells. So there are individual somatic cells which lose part of the genetic material of the organism. Now this is really a very fundamental difference from what we learn in developmental biology. We are used to thinking of development as a process where you go from a single egg to the great diversity of cell types in the organism using the same genome, but differentially transcribing different sets of genes in that genome to lead to the different terminal cell types that we have in these end branches. And this is exactly what is depicted very beautifully here in this single cell transcriptomic data set from zebrafish. What this is really depicting is the changes that are taking place in the transcription different sets of genes from the same genome. In organisms, however, where you have this programmed DNA elimination, you have the original genome of the fertilized egg that is transmitted faithfully into the germline, but in different somatic cells, you have the loss of significant parts of the genome, including functional genes, leading to somatic tissues that have only a fraction of the original genome. This very striking process has been known for a long time. It has been known for more than a century. It was first described by Boveri. But recently people have started studying it and realizing actually that it is not a single unique rare case where this has happened, but there are actually several examples during animal evolution that seems to have independently evolved this kind of mechanism. So we have examples within the vertebrates, in the lamprey, some species of fish and birds and mammals. We have copepods that do this, plus a few other arthropods, and we have quite a large number of nematodes that do this programmed DNA elimination. So this I think exemplifies quite nicely what I mean by discovering new biology in new species which are not model organisms, but which can serve to uncover perhaps new ways in which biological systems achieve things. This programmed DNA elimination is a process that has been very poorly studied because it is not something that you find in any of the major model organisms where we have the tools to understand what is happening, but it is something that people are beginning to dig into in recent years. And this brings me to the process of regeneration, which has been our interest in the last years, I would say in the last decade. I'm talking here about the ability of animals to regenerate a significant part of their body with many cell types, with a complex patterns after a serious injury. And we see here two classic examples of regeneration in the salamander limbs and in planarians that have been cut into small pieces, and where each of those pieces is in some species able to regenerate a whole new planarian that will grow to give rise to a whole animal. We know that these kind of regenerative capacities are relatively widespread in the animal kingdom. There are some phyla, like the nematodes, which seem to be very poor in regenerating. It's not clear if they have such regenerative capacities at the organ level. But most of the other phyla, most of the other major phyla in the animal kingdom have individual species that have very extensive regenerative abilities, and I'm showing here a sample of these species. The major genetic model organisms, C. elegans, flies, and mice, are very poor regenerators. So this is a process that has been relatively poorly studied until now from a genetic approach. And zebrafish is perhaps the best genetic model system that has extensive regenerative capacities and where things are moving in interesting directions. We have started studying regeneration, leg regeneration in this small crustacean, which is called Pariale hawaiiensis, in which, for other reasons, for our earlier evo-devo studies, we had started establishing in the lab and developing transgenic tools and simple methods for imaging and transgenesis. And at some point we realized that the regenerative processes in these animals could be approached with those tools that we had developed. So here you can actually see a limb that has been accidentally amputated in the lab. We often see these animals often these amputations are the result of cannibalism in the cultures. And what has happened is that this limb has been cut off and has melanized at the end here, which is the kind of wounding reaction that you get in these animals. If we take a closer look, what you see is that this leg, which has been amputated at this point, has this scab which has closed the wound. And a few days after the amputation, we can already see with this gene trap line, with this reporter, that the tissue of the leg has already regenerated and has actually formed a mini leg which is folded up in this S shape inside the old cuticle, the exoskeleton that surrounds this leg. So this limb, within a few days, will regenerate a small mini limb inside this restricted space. And the next time this animal will undergo molting, that new limb will be released, it will be straightened, and the new cuticle will harden, and it will become fully functional. So this is a relatively fast process, and one of the major advantages in studying regeneration in this species is the fact that this is a relatively fast process and these limbs are relatively transparent, so we can actually see what is happening inside. So we have the ability, by mounting these animals, to actually observe the full process of regeneration by live imaging under a confocal microscope. And what you will see here is the distal tip, the distal podomere of this leg, which has been amputated here, in which every nucleus is labeled by green fluorescent protein fused with a histone. So what you're seeing here is all the nuclei in this limb. And if I run this movie, this gives you hours post-amputation, so this is right after amputation. You will see that over the course of a few days, cells are initially moving towards this distal end to close the wound. Then they are relatively quiescent, and then after a certain point they will detach, they will start proliferating and undergoing morphogenesis to generate this mini limb that will, after the molting, become a fully functional limb. So what we can see is that within a few days of amputation, within 4 or 5 days, maximum a week, we have formed in here a fully patterned and differentiated little limb that is ready to be exposed to the world and to start working like a leg. Now, in this system, we have so far focused on 3 very basic questions. The first one is to ask how precise is regeneration in restoring the original pattern, cell diversity, and function that we had in the original limbs in these species. So this is a question of fidelity. We have been trying to address which are the progenitor cells that give rise to the new tissues that are generated during this process. And we have been trying to address to what extent the process of regeneration is really a recapitulation of what is happening during embryonic development, or to what extent there are perhaps different mechanisms that lead to a similar outcome. So to address the first question, Alba Almazan and Mathilde Paris in the lab took a single-cell transcriptomics approach to describe the cell type diversity that we have in the limbs. And later I will show you that Chare Chevrim took a more microanatomical approach to describe the detailed morphology of the limbs and to see to what extent the newly regenerated limbs really are precise or not precise replicas of the original limbs in these species. Now, we know from other species that regeneration can sometimes be perfect, as in the planarians that I showed you earlier, but sometimes regeneration merely restores the overall appearance and the function of the organ without necessarily restoring exactly all the tissues that were there before. And it is well known that, for example, in lizards, the regenerated tail has certain tissues that very much resemble those of the original tail, but the vertebrae and the bony structures in this tail are actually never recovered as bone, and you only have cartilaginous structures that replace them after regeneration. In zebrafish, which is a very powerful regenerative system, again, regeneration is quite perfect of the fins, but after multiple cuts, one begins to see some patterning defects that are appearing. So we asked in our Pariale, first of all, to what extent the diversity of cell types that we had there in the original limb is fully restored. So using single nucleus transcriptomics, we defined the different cell types that are present in the adult limbs. This is a U map of the different cell types that that Alba has found. Some of these cells we can identify using known markers. So we know that this is a muscle cluster here. We know that these are different flavors of epidermis, blood, and neurons. But there are also a number of cell classes that we have not yet been able to identify. And then the next step was for Alba to actually see what happens after regeneration. So once you have cut the limb and regenerated, what is the diversity of cell types that you find there? The way she did the experiment is she compared from the same animals fully regenerated limbs, which you see here in the cyan color, and the contralateral limbs which had never been amputated. So these are the controls of uninjured limbs. And what you can see from this plot is that these look very similar to each other, and I will superimpose them so I don't have you trying to guess if there are any differences. Actually, what we see is that all the cell clusters that correspond to each of the cell types that we can identify in these limbs are fully recovered, and the transcriptional signatures, the transcriptomes that characterize each of these cell types seems to really come back to the same as we find in the control limbs. So from a transcriptional point of view and from a cell type perspective, we find that the regenerated limbs and the original uninjured limbs are really indistinguishable from each other. If we look at the proportion of different cell types, so these are the different cell types that we can identify, and these are the proportions of cells that we have in the limb, for example, of epidermal or muscle cells in the controls in uninjured limbs, which are shown here in shades of yellow or brown, versus the regenerated limbs 2 or 6 months after amputation. We really see that the proportion of different cell types are represented in that the different cell types are represented in the same proportions in regenerated and control limbs. And Chare-Chevrim, as I showed you earlier, focused on the microanatomy of these limbs using scanning electron microscopy, focusing in particular on these very finely structured sensory organs, of which there is a very large diversity and very specific patterns in the surface of those limbs. And what Chare found, I will not go into his data, is that again, those regenerated limbs are indistinguishable from the original Pariale limbs before amputation. So our conclusion in terms of fidelity is that from the cell type perspective and from the point of view of sensory organs, we have a complete restoration of the original pattern, diversity, and function as well. So Chare, in addition to the morphological experiments, did some functional experiments to test sensory function. So we find that Pariale is very interesting in this respect because the new limbs really seem to be perfect replicas of what was there before. And it's an interesting question, I think, at this point to ask, why does regeneration have to be so precise in these species? One possibility is that really the process of regeneration is precisely mirroring the process of embryonic development. is actually using the same set of mechanisms, the same gene regulatory networks, and therefore, since the process is the same, the outcome at the end is going to be identical. And this question, to what extent regeneration mirrors development, has been the topic of a project by Chiara Sinigaglia and Mathilde Paris, who have taken a single limb transcriptomic approach to try to compare the temporal profiles of gene expression during embryonic development and regeneration. So Chiara has carefully micro-dissected the individual limbs across this time course of leg development and across the time course of leg regeneration. This is spanning 5 to 6 days, more or less in each case, and has done RNA-seq on individual limbs spanning this time course. And I will give you a brief summary. We recently put on bioRxiv a first draft of this work, which you can have a look at if you're interested in more details. But very, very briefly, we see very nice transcriptional dynamics. This is what you see. This is the top differentially expressed genes for leg development. And this is a time course from early to late. So we see very nice transcriptional dynamics that characterize different phases of leg development. And if you analyze these samples, you can see that the time at which individual legs were collected during embryonic development is very well predicted by the transcriptomic profiles that you find in these limbs. So this is the first principal component in the principal component analysis, but there are a number of ways in which, other ways in which we have looked at this. During regeneration, the situation is more complicated because in addition to this time course, to this temporal signal, we also have a lot of other signals that come from the physiology of the individual adult from which those limbs were collected. So we have a very strong signal that comes from the phase in the molting cycle in which the adult is at the time of the collection. And we probably have other physiological signals in those individuals which really have a very strong influence on the transcriptomes. So I will not go into the details, but Mathilde Paris has generated a model that tries to disentangle these different sources of variation, the individual variation that comes from physiology and the temporal variation that comes from the time course. And once she discriminates between these and she takes the regenerative component, again, we can see that in those samples, we can find quite a nice temporal signal that correlates with the times in which these samples were collected from the point of amputation onwards. And what you can see here, as you might expect, is the samples that were collected at the time of regeneration. This is before the onset of the regenerative program. This is at 0 times post-amputation. Are similar to the ones we get after the first molt. So this 300 here actually reflects the legs that were collected immediately after the first molt. So the transcriptional profile of these legs seems to return to this same state. Now, we have tried several ways to compare the developmental and the regenerative time courses, trying to see to what extent we can align some phases of development with phases of regeneration in order to see if there are any parallels in these time courses. And the overall conclusion has been that it is really not at all easy to do this. The most simplest approach is to do a PCA where you put both the regenerative and the embryonic samples in the same plot. And what you can see here is that the time course of embryonic development, the variation on this time course is really dominating the PCA, and that all the regenerative samples seem to be clustering together, no matter whether they were collected at the beginning, in the middle, or the end of regeneration. We have tried, besides these very global profiles, to go into different subsets of genes and look more specifically whether some categories of genes might give us more specific information. One way of doing this is to define clusters of co-expressed genes during embryonic development and during regeneration. So again, this is our time course from early to late for embryonic development and for regeneration. And we can see that we can find clusters of many genes that are co-expressed, and some of these clusters characterize the early phases of embryonic development, some the middle phases, and some the late phases, and similar things, similar analysis for the regenerated samples. So then once we have done this, we can ask whether the early expressed genes in embryonic development are the same set of genes or largely overlapping sets of genes as the early genes in the regenerative time course, and so on. And the answer has been that it is really not possible to align the time courses and to associate specific clusters in embryonic development with specific clusters of regeneration. And finally, I will focus on this one example. We have plotted here during the time course of embryogenesis and regeneration genes which are heavily involved in cell proliferation and genes which are involved in patterning. What you can see is that during embryogenesis, there is an early phase of intense cell proliferation, which gives way to a phase where a lot of the patterning genes are expressed. During the time course of regeneration, we see a more noisy pattern, but we don't see this distinction of early versus late phases. We see that many of the genes involved in cell proliferation are co-expressed at the same time as the patterning genes. And this is one of the many examples of the kinds of comparisons that you can do to try to compare embryogenesis with regeneration. So very briefly, the take-home messages is first, I think it is important to look beyond established model organisms to understand evolution and also to discover new biological processes which I think are there and have not been discovered yet, simply because we have not had the tools to do that in unexplored organisms. And focusing on our work on leg regeneration, what we have learned very recently is that leg regeneration in pariale seems to fully restore the leg microanatomy and cell diversity, but that unlike our expectations, the transcriptional dynamics of leg regeneration do not really mirror those of leg development in a very obvious way. And there's still a lot of work to do in this on this topic. We are still only touching the surface. So this is our team a few months ago. Mathilde Paris, Alba, and Chare worked on the Fidelity project. Mathilde and Chiara Sinigaglia have worked on the last part, the comparison of embryonic development and regeneration. And our funding sources have been the EvoCell Marie Curie Network and an ERC grant. Thank you very much for your attention.
Michalis, thank you very much. And I take the opportunity to ask the first question. So I think Michalis is a kind of member of a really big group. So these guys, they are amphipods, right? So if I'm not mistaken. So there's plenty of different species around. So do they all have this regenerative capacity? And if not, what's so special about this species? Why to regenerate in this precise way?
Yes. So I don't think our species is very special within the amphipods. I think, in fact, that most malacostracan crustaceans have relatively good capacities to regenerate their legs. I think the major limitation within the arthropods is molting. Arthropods that have a terminal molt, that have one last molt as adults and will never molt again, clearly don't have a way to kind of benefit from regeneration, because even if they did regenerate their limbs would be trapped inside the old cuticle. The molting is necessary to release the limbs. I think most species that have the capacity to molt throughout their lifetime are actually capable of regeneration. Although I have to say I'm not an expert in the kind of widest survey of arthropods and who can and who cannot regenerate. Yeah.
Thank you. Any questions or comments? Okay.
So, based on the analysis of these expression profiles, could you suggest, could you just address the question, what is the molecular pathway? It's more related to transdifferentiation, to stem cell-based pathway, or to de-differentiation of the cells?
Yes. So this question relates to our third interest, which I did not have the time to talk about now, which is really which are the progenitors that give rise to the new tissues during the course of regeneration. We do not know the progenitors for most tissues, for most cell types, but the little we know suggests that we have different kinds of progenitors in those limbs. So in terms of progenitors for muscles, we have found that these animals have cells which resemble the satellite cells of vertebrates, therefore stem cells that seem to be closely associated with muscles. They sense muscle damage and they would proliferate and contribute to new muscle fibers. In the epidermis, we think that every epidermal cell is contributing to the newly regenerated limb. So there is no special population that is serving as progenitors. Every cell is able to enter the cell cycle, make more and make a small patch of cells that will contribute to the epidermis of the regenerated limb. So those are the two examples where we know the progenitors, and the answers that they give us are different. In one case, it's a stem cell. In the other case, it's the actually differentiated epidermis that does the job. And I think we have a lot to learn more, because obviously there are many cell types in those limbs, and we still have to explore where those come from.
Thank you. I saw that Alex raised a hand, so for online connection. So Alexander, please ask the question.
Thank you so much, Oleg. Michalis, thank you. Beautiful story. Could you say a couple of words about the role of the nervous system in this regeneration? Can you ablate the nerves? Will the limb regenerate?
Yes. So, yeah, this is a very good question. It has been observed in many systems that nerves play an essential role during the process of regeneration, from vertebrates to sea stars to all sorts of systems. In our species, Cara has been trying to address this question. What we see is that if you ablate the nerve, the limb will wait until the nerve regrows, and then it will regenerate. And depending on when you ablate the nerve, you may have different defects. So you could keep ablating the nerve and seeing what happens. We think there are at least two things that the nerve is doing in Pariale, and one has to do with wound healing, and the other one has to do with later phases of proliferation and patterning. And these are at least two distinct processes. We don't have still a very clear idea of what these functions are. But we know that the nerve is necessary. It's interesting to note that in different animals, people have found very different roles for the nerves. In some cases, the nerves provide essential growth factors which are needed for regeneration. In other cases, the nerves simply provide the scaffold for other cells to migrate. and to participate as progenitors in the regenerative process. So nerves can have rather different roles in different systems.
Thank you so much. Super interesting. Thank you.
If no more questions, Michalis, thank you very much. Such an inspirational talk. Again, giving us non-model animal lovers hope and inspiration. Thank you very much again, and then thank our speaker.
Thank you.